Hi everyone. So this last video on nucleic acids is the last video in the biochemistry series of videos from this first part of Unit 1. And this is the end part. It corresponds to the end part of the biochemistry note 4. And this will be the final part of the 3D molecule lab that you've been working on. So nucleic acids are named this way because they were originally discovered as molecules in the nucleus of cells. They were discovered back in 1869 by Miescher. So we've known about these molecules that existed in the nucleus for a long time. And the acid part of the name came from the fact that the first molecules discovered had phosphate groups in them, so they were associated with phosphoric acid. We now know that there are other classes of biomolecules that are similar to the molecules of the nucleus, and so they have been collectively grouped together and referred to as nucleic acids. And these include the molecules you're probably most familiar with, which includes DNA. And you may have heard of RNA, which is quite similar to DNA and related to it. But here are some other molecules listed on this screen that you may not have heard of. You might have heard of ATP before, but I would doubt you've seen the other three. The other three, NADH, FADH2, and NADPH, are important molecules in many biochemical pathways in our cells, and you'll be studying them in much greater detail in the next unit. Here you see the basic structural arrangement of the molecules that make up DNA. DNA stands for deoxyribonucleic acid, and it's a long name, but it really tells you some information about the molecules that make up DNA. And I will get into the basic structures of the molecules that make up DNA in just a moment. But the overall structure of DNA, it is what's referred to as a double helix, which really just means a twisted ladder. It has two sides. The two sides are identical to each other. And the sides of the double helix consist of repeating sugar and phosphate molecules. So the backbone goes sugar, phosphate, sugar, phosphate, sugar, phosphate. And the middle of the molecule, which you might think of as the steps of the ladder or the rungs of the ladder, are made up of nitrogen base pairs. And in DNA, there are 4 different nitrogen bases, and they pair up in a certain order. So adenine always pairs with thymine, and guanine always pairs with cytosine. This diagram illustrates what RNA looks like. RNA stands for ribonucleic acid. So remember, DNA was deoxyribonucleic acid. RNA, being ribonucleic acid, is a pretty similar name. It's just missing that prefix deoxy, and that tells us something about RNA. Deoxy means it's missing one oxygen. And that one oxygen is missing on the ribose sugar of the molecules that make up RNA. And I will get to that structure in just a moment. It's similar to DNA, but there are some major differences. DNA, remember, was double-stranded, put together in the middle by those nitrogen base pairs. RNA is a single-stranded molecule. It's actually a single-stranded copy of one side of the DNA molecule. As I mentioned, it has a different sugar. The sugar is called ribose instead of deoxyribose, but it does have that sugar phosphate backbone to it. The nitrogen bases just kind of stick off of that sugar phosphate backbone, and you might notice that compared to the previous slide, one of the bases, thymine, is not here. Instead, there's a base called uracil. So uracil is right here. And that's one of the differences between DNA and RNA. Another difference is that DNA is confined to the nucleus. It's a really large molecule, so it cannot fit out of the nuclear pores in the membrane around the nucleus in order to get out to the ribosome where proteins get made. Instead, this single-stranded RNA molecule will be made off of the DNA, and it's small enough to leave through the pores of the nuclear membrane, go out to the ribosome, and be decoded into the protein that the DNA coded for. Now, DNA and RNA are polymers. They are these long chain molecules made up of repeating smaller molecules. Those repeating smaller molecules, or monomers, of DNA and RNA are referred to as nucleotides. Nucleotides contain 3 parts. They have a phosphate group, a deoxyribose sugar, and a nitrogen-containing base. So we see that here in this very simple diagram with our phosphate, a deoxyribose sugar, which is a 5-carbon sugar, so it's referred to as a pentose sugar, and a nitrogen base. And in DNA, those nitrogen bases will be adenine, thymine, guanine, and cytosine. In RNA, we see adenine, uracil instead of thymine, guanine, and cytosine. Those are the different nitrogen-containing bases or nitrogenous bases that are found in these monomers. And remember that the phosphate and sugar make up the backbone of DNA, and the nitrogen bases make up the rungs or the steps of that double helix. Now this is the biochemical structure of a nucleotide. Instead of showing it in a really simplified form, here you can see all of the atoms. So the phosphate group is a highly charged, extremely polar, very negative group on the nucleotide. It has negatively charged oxygens. And it's attached to this 5-carbon sugar called deoxyribose in DNA. And in RNA, remember it was ribose. So it was simply missing an oxygen. I think it's on one of these OHs that it's missing an oxygen. Here's one of the nitrogen bases shown right here. I'm not sure which one this is. I think this would either be A or G. That's either adenine or guanine. I'd have to look it up, but I know that adenine and guanine have 2 rings of carbons. And so this is one of those 2. And you can see it's a nitrogen containing. There's a lot of nitrogen in this molecule. So hence the name nitrogen base. So here are the 5 different nitrogen bases in their structures. And I mentioned that adenine and guanine had the 2 rings. So you can see in this diagram right here, whoops, let me find somewhere I can write. Here we go. The purines, this is fun. Here we go. The purines, they have the 2 rings, the double rings. So they are double ringed. And the other 3 nitrogen bases, cytosine, uracil, that was the one in RNA, and thymine are all single ringed. And they're referred to, whoops, let's get that back. They are referred to as pyrimidines. And this can be pretty tough to keep straight. What are the purines? What are the pyrimidines? Well, one of the ways that I've tried to remember this when I was struggling with this myself is that if you look at the word purine, it has the letter U in it, and the word double also has the letter U in it. If you look at pyrimidines, it has a lot of I's, and the word single has an I in it. So I don't know if that will help you at all, but I always try to come up with these memory devices or mnemonic devices to help me remember patterns in terms and certain facts. So purines are double ringed, pyrimidines are single ringed, and you might be wondering, why do I have to know this? Well, it explains the structure of DNA, and only when the researchers that are credited with the discovery of the structure of DNA are Watson and Crick, and that's a really interesting story on how they came up with the model, the structure. It's based on some other people's research as well, but they won the Nobel Prize for their work. Once they knew that adenine and guanine were double ringed and that the other 3 were single ringed, only then could they figure out that adenine had to be chemically bonded to thymine and guanine had to be chemically bonded to cytosine. It was the only way that that molecule could exist, and it's related to how many rings are in the structure. So it is important to have an understanding of the structures of these 5 nitrogen bases. Now, of course, I will never ever ask you to draw these from memory, but you should just be familiar when you see them that you go, oh, okay, that's a nitrogen base. So moving on from the nucleic acids that are related to the nucleus, which are DNA and RNA, it brings us to this very important nucleic acid called ATP. ATP stands for adenosine triphosphate. And you may have heard about this molecule in your grade 11 biology studies because this is our energy molecule. When we have extra energy, for example, after you've eaten a meal and you have some glucose available for your cells to burn, you will convert that energy and store it in bonds in this bond right here, in this molecule right here called ATP. So adenosine triphosphate has an adenine group. It has a ribose sugar, just like we saw in RNA, and then it has these 3 phosphates. And this is really the key to how ATP works. You may want to draw the structure into your note. This bond that you see between the phosphates contains a great deal of energy. So when we have energy available, we will add phosphates onto a smaller molecule and create ATP. When our cells require energy, we break the bond here between 2 phosphates and we release that energy so our cells can use it. So I've often heard of ATP being referred to as the energy currency for the cell. So think of it this way, it's like money for your cells. When you work your job and you get a paycheck and you make money, you store it in the bank. You save it in the bank. And when you need it, use your bank card and your debit card and you spend that money. Well, this is what we do with energy and this molecule ATP. And I'll show you the other molecule that we convert in order to make ATP. So this diagram shows the hydrolysis reaction of ATP in order to form a very similar molecule called ADP. And you can probably guess from its name, adenosine diphosphate, that it has lost a phosphate. So we start here with ATP, and here's our adenine group right here, and our ribose sugar, and then these critical 3 phosphates right here. And remember, there's a lot of energy stored in the bonds between these phosphates. Your cells will perform a hydrolysis reaction, which involves putting water into the reaction. It's the opposite of a condensation reaction, and that will break off a phosphate. So here's a phosphate coming off. And with that, we release a lot of energy. The little caption right here tells you that this conversion produces about 7.3 kilocalories per mole of ATP. And kilocalories per mole is probably not a unit of measurement that you're very familiar with, but in the processes of cells, that's a lot of energy. This next nucleic acid is called NADH, and that stands for nicotinamide adenine dinucleotide. Now don't worry, I don't expect you to remember these long form names. Every scientist that works with this stuff uses the short forms. Everybody calls this NADH. They don't call it by its long name. So I'm just giving you the long names to show you what the letters stand for. NADH is a really important energy carrying molecule in our cells. All living things have this molecule. And it's not that important that you understand its reactions right now, but you will see it in the next unit when we do metabolic processes and cellular respiration. And you'll see its important role that it plays in transferring energy throughout the cell. But it just it involves an oxidation and a reduction reaction, which we'll be going into again in the next unit. This molecule is called FADH, and when it becomes reduced, it is called FADH2. So when it gains hydrogens and electrons, it's called FADH2. It stands for flavonoadenine dinucleotide. It's like NADH in that it's one of these energy-carrying molecules that helps to move energy around the cell when metabolic processes are going on. So when you are converting glucose into energy and making ATP in your cells, NADH and FADH2 are important energy carriers to help that happen. And again, this is all new to you. I'm just introducing it to you. I don't want you to worry about it too much. In the second unit, we will look at this in much, much more detail. For now, if you can just remember that ATP, NADH, and FADH2 are examples of energy-carrying nucleic acids, that's perfect. So I just want to end off the video with this last nucleic acid. This one is not found in us. This one is found in plant material, in plant cells. It's NADPH. NADPH, very similar to NADH, but it has a phosphate in it. So its name is nicotinamide adenine dinucleotide phosphate. And you can see the phosphate group here. This is a nucleic acid that's used by plant cells to move energy from one metabolic process to another. So very similar role to our NADH and FADH2, but again only found in plants. So this brings us to the end of biochemistry. Now, not the end of the biochemistry unit, but the end of part 1 of our biochemistry unit. We've started with a very general look at overall basic chemistry, basic biochemistry, um, ion structure, atomic structure, isotopes. We looked at the role of electronegativity. We looked at bonding models like ionic and covalent bonding, polar versus non-polar covalent bonding. We looked at intermolecular and intramolecular forces and how those were different. We looked at what a buffer is and how a buffer works. We looked at the special properties of water. And then our focus has been, we moved in and spent a lot of time on the 4 macromolecules: carbs, lipids, proteins, and now nucleic acids. When you're thinking about where should I focus my energy for this part of the unit, it's those 4 macromolecules that I want you to focus on. Those are the basis of our cells. That's what our cells are made up of. Those are the molecules that become our building blocks that we build up and we tear down and recycle constantly in all of our cells. They're the molecules that keep us alive. So focus your energy on understanding the structures and the functions of the various carbs, lipids, proteins, and nucleic acids, and you're good. So good luck with all of this, and keep asking those questions of me. Think about making study notes. Think about trying to approach this in a way that works well for you so that you can learn this material even better than what you know now. So bye for now.